In this video, we'll be talking about the Hardy-Weinberg equilibrium and how selection violates it. Population genetics studies the variation of a population over time, and it does so using the Hardy-Weinberg equilibrium. The equilibrium serves as our starting genetic parameters given the assumptions listed here. So, if all these assumptions are met, then we can find our genotypic frequencies using this equation. You'll notice one of the assumptions is that there is no selection occurring. So if we come across a population in which selection is occurring, we'll have to alter the equilibrium to account for this. Let's look at this population of 70 stars. In this population, we have three distinct phenotypes. We have plain stars, which are homozygous dominant, we have pattern stars, which are homozygous recessive, and we have shiny stars, which are heterozygous. Now this population is currently in equilibrium, and we can confirm that by finding and using the allelic frequency to find the genotypic frequency, and then using that to conduct our chi-square test. Now, while this population begins at equilibrium, we're going to observe selection on it, which we discuss in terms of fitness. Fitness is the ability to survive and reproduce, usually represented with the letter W. Looking at our population, although we have 70 stars, not all of these will survive long enough to reproduce. In fact, these stars are prey to birds, and these birds have preferences. Because of this, the shiny stars are more attractive to the birds and thus more likely to be eaten before they can reproduce. We can quantify this selection using a fitness number between 0 and 1. In this table, we can see our plain stars have a fitness of 0.795, our shiny stars have a fitness of 0.425, and the pattern stars have a fitness of 1. With these fitness values, we see that our higher fitnesses are pushed outwards towards our homozygous genotypes, whereas our lowest fitness is the heterozygous. This type of selection is called disruptive selection because it favors our extremes, and we'll see this more clearly in a minute. With this information, we can multiply the fitness values by the initial frequency of each genotype to get an unadjusted survivor rate. This tells us the proportion of our original population that survives after selection. In order to predict further generations, though, we want to find the relative frequencies, and we'll do this by calculating the average fitness. The average fitness, or W bar, is simply all of the unadjusted survivors added up. So in this case, we'll get 0.6698. Now with this number, we can divide the unadjusted survivors in order to give us our relative frequencies. Now, if you compare our initial frequencies to our frequencies after selection, you'll notice the frequency for heterozygous shiny individuals has decreased, and the frequency for our two homozygous groups has increased. Over time, this disruptive selection will push our phenotypic traits to the extremes, and we'll observe a bimodal population like this.